I thank the organizers also for this wonderful deep dive in the past. Can you put, yes. Sorry. So we have been awarded a group of 4 people, myself when I was at Columbia, Hiroyasu Nakano from Toho University in Japan, George Mosialos from Thessaloniki in Greece, and Guri Ghosh from USC SD. So we have been awarded the Early Career Grant back in 2001 about signal-dependent regulation of NF-κB activity. So the question is, and that's how I want to start, what is NF-κB? We may have a problem here. Doesn't change. Oh, yeah. So NF-κB is a pleiotropic and the most powerful transcription factor in mammals. Normally it's held in the cytoplasm by IkB proteins, but it rapidly translocates to the nucleus upon cell activation. The NF-κB system is evolutionary conserved from Drosophila to, excuse me?
This isn't the right one. There should be another one up there.
Oh, there is another one? Yeah. There is no other one. Oh, yeah. Is this one?
Yeah,
okay. Amazing. Okay, great. And how it moves forward? Of the big green button. Okay, great. So, the NF-κB system is evolutionary conserved from Drosophila to humans. NF-κB senses and responds to cytokines, pathogen, stress, etc. NF-κB plays the most important and evolutionary conserved role in actually in the immune system and beyond that. Dysregulation of the NF-κB system can cause many diseases. For example, cancer, metabolic and developmental diseases is the focal point of various signaling pathways, and it is one of the first oncogenes. So the activation of NF-κB is a remarkably complex biochemical process. So here you see some a very simplified version of that. So when a receptor is activated, in this case is the TNF receptor, then there is a signal transduction pathway cascade which activates a kinase complex in the cells called the IkK complex, which phosphorylates the inhibitor. The inhibitor is called IkB, which upon phosphorylation, this signal phosphorylation gets ubiquitinated and then is distracted by the proteasome, and the free NF-κB translocates into the nucleus and activates gene expression. Now, NF-κB receives a huge variety of signals and then responds in an even large, and then it activates or represses an even higher number of genes. Genes which are related with the immune system, as I said, and genes also that are related to cell survival, but also to apoptosis. So there is a balance between cell survival and apoptosis. And our team, which we put together 25 years ago, actually addressed this question. Now, NF-κB is 5 different proteins. 3 of them, REL-A, REL-B, and CREL, contain a transcriptional activation domain. All the NF-κB proteins bind to DNA via an amino terminal domain, this green one, called REL homology domain. This is from the oncogene REL, which was first identified. And then there are also, and they can form different kind of homo and heterodimers, and the IkB proteins are 6 of them, you see them all here, are all characterized by ankyrin repeats, which are protein-protein interaction domains. As I said, the rel homology domains, I mean NF-kappa B proteins can form different kind of homo and heterodimers, and all of these have activity, and the activities of all of these is regulated in a stimulus-responsive manner and in a cell-type specific manner. And they also have distinct transcriptional activation activities. Now, one of the remarkable things in the NF-kappa B system is that it has embedded many negative feedback loops and also some positive feedback loops. And the very first one discovered a long time ago was that NF-kappa B, when it's activated, when it's turned on, activates the expression of its own inhibitor, of the IkBs B alpha gene. And as I'm going to show you in a while, it can also activate other inhibitors which turn off activation, which is very critical to maintain homeostasis in the cell. In fact, if you look the entire pathway of NF-kappa B activation, NF-kappa B regulates, negatively regulates nearly every single step in this activation. So it's a remarkably complex system which has embedded many negative and some positive regulatory loops. So a member in our team, George Mosialos, actually discovered that one of these negative autoregulatory loops is a gene which causes benign tumors in humans. In fact, these are formed in the forehead, and those are called cylindromas, and the gene is called cylindromatosis tumor suppressor gene. As you see mutations there. So the thing about this gene is that it encodes a de-ubiquitinase. It encodes an enzyme that removes ubiquitin, the ubiquitin chain from other proteins. And let me show you, it's an animation of 3-4 slides, how this thing works. So, imagine an inducer, TNF binds to its receptor. This causes a trimerization of the receptor. The cytoplasmic parts are brought together, and this causes the recruitment of various adapters and ubiquitin ligases. Now, next, when this happens, then one of the ubiquitin ligases, this one, CIAP2, adds ubiquitin groups in several components of this receptor system. And now this actually causes now the recruitment of other proteins, kinases, and other adapter proteins, which are recruited via the ubiquitin chains. Now, the NF-kappa B system was the very first system where it was shown that ubiquitination actually can work as a signal like phosphorylation works, like a regulatory signal, not only a distraction, but also a regulatory signal. So when this kinase is recruited, this kinase, then another ubiquitin ligase is recruited, and this causes the very first recruitment of the IkK complex. The IkK complex is the one which phosphorylates IkBs B, and this is followed by the acetylation of the further, not acetylation, ubiquitination by Lubach of all the components in the receptor complex. Now, this is the best substrate for very efficient recruitment of the kinase complex. As I said, there is some initial recruitment from this ubiquitination here, but here there is another ubiquitination which is more effective. When many molecules of the IkK complex are found associated with the receptor, then there is phosphorylation of the IkK, autophosphorylation, and direct phosphorylation from TAC1, which leads to the activation of the IkK, which in turn IkK phosphorylates I-kappa B. I-kappa B, now the phosphorylated I-kappa B is the beautiful substrate for ubiquitination, and this is the perfect food for the proteasome. And then the proteasome removes I-kappa B alpha, and then NF-kappa B gets into the nucleus and activates expression. One of the genes which is activated is CYLD, is this de-ubiquitinase I told you. And then CYLD is produced, and then de-ubiquitinase everything here, and this causes destruction of the complex and termination of signaling. So next, what our team investigated was, you see, this is a simplified view of all these interconnected cross-talking pathways of NF-kappa B with other similar or quite different pathways. So what we investigated, Hiroyasu Nakano, was like how TNF can actually induce NF-kappa B to prevent cells from dying, so inducing cell survival, and at the same time blocking apoptosis, because TNF induces separately junk. Junk is a kinase called Jun N-terminal kinase, which is very well known to induce caspases and which induce apoptosis. So what Hiroyasu found was that actually when NF-kappa B is activated upon TNF-alpha, then represses reactive oxygen formation, which is required for the activation of junk. So that's how the whole pathway stops here, and you don't have apoptosis. Similar, NF-kappa B via an unknown mechanism stabilizes the protein C-flip, which actually destroys caspase 8, and this makes the cell surviving. So you see how complex it is, and this actually could be different from cell type to cell type, and can also be different from inducer to inducer. The next thing we investigated, our team, was another very weird characteristic of NF-kappa B, which is that it regulates most of the cytokine genes in a stochastic manner. What stochastic means that when NF-kappa B gets into the nucleus, then does not activate in every single cell all the target genes, but there are genes which are but there are cells where the thing has been activated, and then there are cells which it doesn't. And in fact, this stochasticity is caused, we showed, because NF-kappa B is a limiting factor. So what we found in the context of investigating this was that it should exist a mechanism which actually drives or helps NF-kappa B to find its targets. So what we found was, and I don't have the time to tell you how, is that NF-kappa B is initially received in the nucleus by 3 different sites, which they are called NF-kappa B reception centers. NF-kappa B is received there because it binds cooperatively with another protein called TH-POC. And then when it binds there, TH-POC oligomerizes in the nucleus and then brings together loci that are in which there are targets of NF-kappa B, like the interferon beta locus. You see that here. And this is how NF-kappa B is delivered to the target genes. So basically, in this case, those NRCs work like Volt or like eFood, delivering stuff. And this is how NF-kappa B finds its target genes. And this, because these interchromosomal interactions are highly are highly stochastic, as we know, this occurs only in 20% of the cells, and that's why only about 20% of the cells express on the average any NF-κB target gene. And this is how it works. So NF-κB and TH-POC bind together to the NRCs. TH-POC is oligomerized and brings together the target genes. And here you see this in a beautiful picture where you see this interaction, the green and the red NRC and the interferon gene is the site where RNA is produced, which is the yellow color. And in fact, this doesn't happen for each gene separately. There are groups of genes which are all recruited in the same place. So those are hubs which are being formed, and those hubs allow all these genes to be expressed in a stochastic manner, but in the same cell. And, okay, and I'm finishing with 2 or 3 slides about the structure of NF-κB. So there are many things which really surprised us 25, 30 years ago when we started investigating NF-κB. One of the things was its unusual high affinity for the DNA. An average mammalian transcription factor has an affinity for DNA which is about 10 to the minus 9. NF-κB has about 10 to the minus 11, which actually approaches the number for bacterial proteins. For example, the lac repressor binds to 10 to the 12 or 12 to 10 to the 13th, which is a remarkably high number. So, and in fact, this was explained when the crystal structure of the NF-κB was solved. And as you see here, you look, the DNA is perpendicular to the screen. So as you see, NF-κB actually encircles the entire DNA, making both sequence-specific and sequence-unspecific contacts. In fact, different heterodimers and homodimers have different affinities for different kinds of sites. And here you see an optimal P50 homodimer site, which is 11 base pairs, and the optimal relay binding sites, which is 9 base pairs. And if you look and you investigate very, very carefully, you see that each one of the heterodimers and the homodimers have different optimal binding sites, a different size of optimal binding sites, etc. And in fact, the other very strange thing was that NF-κB also can make, as I said, high affinity. Remember that, binds with high affinity. But if you mutate one of the sites where one of the 2 monomers in the dimer bind, yet it can still bind. And this increases tremendously the number of potential binding sites in the genome. That's why it's limiting, because it binds in many, many different sites. Some of them do matter, and some of them don't matter for activation and gene expression. And the second thing was, and this is my last slide, that depending on the sequence, NF-κB can induce different kind of distortions on the DNA. This is called DNA bending, and it has been shown that DNA bending plays a crucial and a very important role in cooperative DNA binding and in allowing the formation of higher order nucleoprotein complexes. And this is the group, this is now. We were much better 25 years ago, okay? So this is me, this is Hiroyasu Nakano, this is George Mosialos, and this is Guri Ghosh. And as I said, this was an early career award, 2001, 2004, and we thank HFSP for this opportunity. Thank you.
We have time for a couple of questions, one by my colleague here.
Let me ask you the first question. Your grant dates back to 2001, which was the very year when we introduced the Young Investigator Grant, now called Early Career Research Grant, which is great. That's why I selected you actually to give a talk. To what extent, I'm curious, to what extent do the four of you still collaborate?
Okay, that's a tricky question. I would say yes, we collaborate. The truth is that we don't collaborate much. I collaborate with one of the guys, this one, because it's much easier because we are in the same country now. But we don't collaborate. But this meeting, which again, gave us the opportunity to talk and they to explain me what they are doing because I'm here to actually present it. So gave us a lot of ideas and to plan a new collaboration on a different ground, of course.
Good, great.
That's a very good question.
How it should be. Another question.
Yeah.
I was wondering if you can predict which cell will respond stochastically to produce interferon beta.
No. Otherwise, will not be a stochastic thing, right? In fact, do I have 1 second, Mr. Chairman? Mr. Chairman, can we go back in 3-4 slides? How this thing goes back, does it? Go. Oh, oops, one here. No, one backwards. Here, okay. I didn't have the time to show you this experiment. This is a beautiful experiment suggested by a friend. So how you prove that something is stochastic, right? So here what we did, this is a microtiter plate. So we took cells, single cells, right? And we put one cell in each well, right? And we allow the cells to go from 1, 2, 3, 4 generations. So if stochasticity is not a genetic thing, or okay, let me say it the other way around. If stochasticity is genetically determined, then you expect that if, for example, this cell, which does not express the gene, when it will be divided for a long time, all the cells would be negative. But this is not the case. So you see, from a negative cell, you can get after some time, you can get all the positive things. So because it is calculated that in this case, 1 every 5 cells will express. The weird thing, which we cannot yet explain, is why this is defined 1 out of 5. Because it doesn't matter how many times you do the experiment, always is 1 out of 5. So something is tricky here. We are working on that. Great question.
All right, thank you again.
Thanks.